My name is Jennifer McCright, and I'm also a student with the University of Washington Engage program. As a graduate student, I get really specialized training in how to do a very specific type of research. The Engage program gave me another type of training that graduate students rarely get. I learned how to talk about my science in a way that is engaging, enlightening, and informative. You can learn more about our innovative program at engage-science.com. And now to our second guest. Shivani Gupta is a fourth-year PhD student in bioengineering. Her interests include proteins, stealing ideas from nature for cool new biotechnology, reading, and for those precious leisure moments, skiing, ballroom, swing, and salsa dancing, hot yoga, and, if lucky, traveling. Please give a warm welcome to Shivani Gupta.
Thank you, Jennifer, for that very kind introduction. So today I'd like to talk about the keys to better medicine. And by keys, I'm referring to two things. One is very metaphoric, that is a solution or a clue to help build better medical technologies. And the second is actually quite literal. I will in fact talk about keys that can help us make better medicine. So to do this, I'd like to start by introducing you to a tiny world, a world that we're very familiar with now after Stella's wonderful talk, a world full of workers, many machines and tools, much like our own, but within all of us and every living creature. And that's the world of a living cell. Every cell is composed of every living creature is composed of millions and millions of little cells. Our own human body has some 37 trillion cells. Even this adorable little baby is made up of trillions of adorable little cells. And each of these cells have many different responsibilities in maintaining our body, such as fat cells, which help store energy, brain cells, which send and receive all kinds of information about what we're doing or what we're thinking, muscle cells like those in your heart that provide movement, and red blood cells that carry oxygen throughout our body. And these are just a few of the many types of cells that help us do what we do. But how do these cells do what they do? How do these cells work? Well, cells use tools and machines called proteins to accomplish their jobs. We're now very familiar with proteins, but maybe you still think of proteins as eggs or chicken or meat, and you'd still be right. When we eat these foods, our body breaks them down into their many proteins, and then those proteins into their individual building blocks. We then take those building blocks and rebuild them into the sort of proteins that our body can use. These proteins can include things that are called transporters, move things around in our body. They might provide structural support, they might break things down, they might help speed up chemical reactions in the body. They might even help identify intruders like viruses that enter the body. So proteins are the little machines that the cells use to get things done, but let's be really curious and let's ask, how do proteins do this? How does a little protein interact with all sorts of other things? We heard about MinC and all these other very interesting proteins in bacteria. How do they work? Well, proteins actually bind to things. And when I say bind, I mean something like what a key and a lock do. Here you have a key binding to a very specific place in a lock. Then something happens. A door opens, a box opens, your car starts. Basically, that key binding to a specific place in a lock leads to something happening. And in much the same way, proteins interact with other things by binding to them specifically. It can bind to this chemical and move it around places. It can bind to another protein, and if it's bad, maybe mark it for destruction. Or it can bind to a surface. And you'll notice in each of these cases, the protein binds to a very specific place, much like the grooves of a key bind to a specific place in a lock. So I want to talk about a really specific protein, and that's an antibody. You might be familiar with an antibody as something that binds to anything foreign in our body, like a germ. Our cells actually produce antibodies as a defense mechanism to fight anything foreign that enters our body. It tags it as bad so that we know to begin to fight it. And again, the way it does this is by binding to it like a key to a lock. And just as only one type of key can open a certain lock, only one type of antibody can bind to a particular germ. That uniqueness actually makes antibodies incredibly useful for medical technologies. Just think about the many ways we use keys and locks in our everyday lives to protect us, to hide things, to start cars or machines, to make sure only the right people have access to the right places. So in much the same way, proteins are incredibly useful. And you might ask why antibodies are incredibly useful, and you might ask why antibodies and not just any other protein? Well, unlike other proteins that do bind things, antibodies are actually very conveniently made from mammals. Just like our bodies produce antibodies to anything, mammals also produce antibodies to anything that they are exposed to. And so engineers have taken advantage of this. It's all the rage right now because antibodies can help us make sure that the medicine attacks just the bad guys in our body. In other words, it could potentially help reduce side effects. So how? Let's say we want to make some new medicine that binds just this blue germ. We just take this blue germ and we inject it in some small animal like a mouse. The mouse's body then produces antibodies against the blue germ because it knows it's bad, it knows it's foreign. And we can take those antibodies and use them to make a better, new, targeted drug that just attacks the blue germ. Very useful. Some of you might say, well, these are really adorable animals I'm showing you here, and that's kind of sad that we're doing that, but I'm going to ask you to hold on because I'm going to provide an alternate. So I also want to share another way that we use antibodies every day, and that's in diagnostic tests, or tests to diagnose illnesses. For example, many companies are trying to find better ways to fight HIV, to diagnose HIV. The way they do this is by first taking a blood sample from a patient. In that blood sample, you'll look for this little HIV virus particle. And to capture that HIV virus particle, you'll use this diagnostic test. As you can see, it's just a strip coated with antibodies. And these antibodies will only bind HIV virus particles. So if we take all of that stuff from the blood and we just flow that across this test, only the HIV virus particles get captured. Everything else just washes away. And so we can now detect just the HIV virus particle. So since antibodies are so incredibly useful, engineers are trying to produce them for more and more applications. But while our bodies are really good at making them for our own use, it's actually quite expensive and time-consuming to make them for medical technologies. Plus, relying on these animals to produce antibodies can lead to a lot of inconsistencies. And lastly, it's very difficult or even impossible to control binding to targets when you use an antibody. You can't control when it binds, and it's very difficult to force it to release a target. So given how useful these natural locks and keys are, it's quite a drawback if we want to use them to do great things in medicine. So what do? Well, remember that an antibody is just one kind of protein that acts conveniently like a lock and a key. But maybe instead of using an antibody, we can use a different protein that kind of looks like an antibody already, but is much less expensive and much easier to engineer for our own uses. So that's what my lab does at the University of Washington. We're working on building a better antibody using an entirely different protein. Now, the protein I'm talking about comes from a cell from a very different creature, but one that you've seen today. And here's our little friendly guy, Mr. E. coli. Again, this is the harmless bug that's found in our gut, and you've mostly heard about the bad kind from bad spinach, but as Stella's mentioned, E. coli have a lot of benefits as well. So just like in animal cells, bacteria cells also produce proteins to help it survive, as we now know. One group of proteins that this bug produces form a long appendage, much like an arm or a leg. It makes hundreds of these to help it attach to surfaces. These are called fimbria, and E. coli use fimbria to stick to the gut, or to any surface that it likes. So how do these arm-like fimbria stick to things? Well, you'll notice at the very end is something that's kind of like a sticky end. The sticky end is its own little protein, and this is the one that looks a little bit like an antibody. This protein sticks to little sugars that can be found coating our gut and many of our body cells, and that's why E. coli can live in our body, because it attaches to those little sugars. So why are we picking this protein that comes from bacteria of all things? What makes it special? Well, this protein does not bind to things unless it's triggered or activated to do so. It stays in this no binding state where the target can't really fit into the end. But if someone pulls at it, it switches to an activated state where the target does fit into the end. If that sounds complicated, just think of a Chinese finger trap. Maybe when you were a kid, someone tricked you into trying a Chinese finger trap and you realized you were stuck. The harder you pulled, the harder the Chinese finger trap stuck around your finger. But if you loosened your force on it, the Chinese finger trap released. Well, basically, force is making the bond tighter around your finger, whereas less force is releasing the bond. And that's kind of how this protein works. So what does this mean? It means that we have a protein that can be controlled or controlled to bind to its target, or controlled to release its target. In this case, we're using force just to activate it. So using this protein, we can make antibody-like proteins that are activatable. We like to call these activatable antibodies actibodies. So how would one use them? Well, in the same way that a new antibody key can be made for any lock that interests us, we can make a bunch of actibodies that each bind to something different, each an individual key for whatever lock we like. How would we do that? Well, unlike with animal cells and antibodies, these bacteria cells won't just magically produce actibodies to anything we expose it to. But that's okay. Instead, we'll use the building blocks that make up these proteins, and we'll do the engineering ourselves. Maybe when you were young and you were playing with Legos and you lost the instruction booklet, you still tried to piece Legos together. You were not sure what you might end up with, but you knew if you put some of the right pieces together, you'd have something useful. And that's basically what we're doing. We're putting different pieces, different building blocks of this protein together, and by manipulating them, we can end up with a bunch of different actibodies that bind to something that interests us. So now, imagine being able to pick and choose between 1 million versions of actibody proteins, 1 million different keys. One might bind to HIV virus, one might bind to a blood cell, one might bind to a flu virus. You can think of it, in fact, as a library of actibodies. We can scan through them for the one that fits our application. And remember, this library of actibodies are a huge step better than those old antibodies because they're activatable. We can trigger them to bind to their targets or trigger them to release their targets. But what do I mean by this trigger? Well, I kind of talked about the natural trigger of the protein, that's force. Something pulling on it causes the protein to switch to its binding state. But force can be kind of an abstract concept, kind of hard to control. It's not very convenient for a lot of applications we want to use this in. Fortunately, we can engineer our actibodies to react not to force, but to maybe some small something by binding to a specific place right in the middle of the protein. By wedging itself in there, the small molecule acts kind of the way force does, switching it to a way that it can actually bind to its target. That small thing can be another antibody, it can be a chemical like zinc or calcium. These are much easier to work with because we can just throw them right in there when we need them. So let's regroup for a moment. I've told you all about this exciting new binding protein, this actibody. I've shown you how we can make a whole library of them and the sort of triggers we can use to control them. You're probably thinking, well, Shivani, that's fascinating, but why would this be useful? How would you use an actibody and its trigger, and why is it really any better than an antibody? So to answer that, I want to take us back to our diagnostic tests. Remember, in a diagnostic test, engineers use antibodies to capture any germs from a blood or a saliva sample. For example, that sample of blood, when you flow it through here, germs are captured, as you've seen before. Now, for detecting the germs, it takes a large volume to pull these germs down. Somewhat inconvenient, but in this case, since there are so many, they can be detected easily, as you can see by that vivid purple. What if there are not enough germs to detect? In many cases, especially at the early stages of a disease, the germs that cause that disease might not be very concentrated. In fact, there can be so few of them that even if antibodies capture them, they're too few to be detected by most tests that we have. For example, as you can see here, in the large volume it takes to pull down their germs for detection, the few germs present are way too dilute to detect. You can barely see the change in color. And that makes it difficult to diagnose diseases early on when it's really important to do that. If, however, we can both capture and release the germs with just a simple trigger, we can increase the concentration of the germs in our little sample so that they're easier to detect. We don't need that large volume anymore. So let's take the sample that has just a few germs in there, too few to detect out of that large volume. But you'll notice instead of antibodies, we now have actibodies. So we'll flow this across, it captures those few germs present. But now we'll flow through our trigger. And this trigger, I don't mean force, but that's something small, like zinc. This is in a much smaller volume. A large volume isn't needed now because we have a trigger to do the job instead of releasing the germs for detection. So as we flow the trigger across, the actibodies are switched to their no binding state, so the germ no longer fits. Now in that smaller amount of liquid, the germs detach from their antibodies and suddenly they're much easier to detect. And that's because the concentration is much higher in that small volume. So what does this mean? Well, just by using our actibodies, our activatable protein, we can maybe diagnose illnesses at earlier stages. We can maybe improve current diagnostic tests without needing fancy, expensive equipment. So this is pretty cool for diagnostic tests. And the impact goes beyond that. I mean, you might wonder, how would each of you really benefit by engineers like us using actibodies instead of antibodies? Well, for one, they're way less expensive and faster to produce than antibodies. And that's because they're made from bacteria. As you might know, bacteria love to grow. They grow really quickly, they'll double in 15 minutes or less, and each time they double, we've programmed these bacteria to make our actibodies. So it doubles, we have more actibodies. It's fantastic, it's quick, and what that means is that could maybe reduce the cost of our our medicine and our diagnostic tests, and I'm sure we can all get behind having cheaper healthcare. Secondly, we have that added control that makes our brand of keys, our actibodies, way more useful than traditional antibodies. We can control binding and release of targets. So if we make this up to scale, this could have huge impact on diagnostic tests, as I mentioned, on how we deliver medicine and how to avoid side effects of medicine. So it could really have a huge impact in the field of medicine. So that's our big picture. I've shown you all these grand things that we hope we can do with the actibody, but we still have a ways to go in my group. We've built our library of actibody proteins and we're currently working on scanning through them for the ones that interest us. But I wanted to share this big picture with you because that's what motivates us and that's where we hope we're headed. So with that, I'd like to thank all my colleagues in my lab, including my wonderful mentor and advisor, Wendy Thomas. I'd also like to thank Shelley and Juliana from the Engage program for all they've taught me and the great opportunity to speak here tonight. Without their help and without all my classmates' help, this would be an incredibly technical and boring presentation. And lastly, I'd like to thank these funding agencies for making what I do possible and making the Engage program possible and letting me basically do what I like to call risky science. So I'll take your questions. Thank you.
I have two questions. Excuse me. The first is, are there activatable antibodies in our body now?
There aren't really designed to be activatable in our body right now. Their main job is just to bind to things. So I would say no.
Just taking all these things that people take to, supposedly increase their resistance, if that has any effect, but apparently not. The second question is a little bit more philosophical, and I'll sit down for the answer. I'm imagining years from now, a pill that you take that has this library of activatable antibodies that protects you from everything. And thinking of books like Guns, Germs, and Steel, where you have populations that die because they haven't got the resistance that some other population has. But now we have this world where we all have the same resistance. Just curious, what might be the implications in terms of human evolution? Big question, but I'll answer.
Definitely a big question. So I don't believe the goal of this project is to make something that injects us all with a library of things that will make us resistant to everything, because as you might have heard in media, having high resistance means something will eventually become resistant to you, to the antibodies that you've built. So that I think that's just an ongoing thing. Things will evolve to continue to fight you. So I think that's an ongoing battle. I don't know if it will be an end-all solution. Definitely a big question, though, to think about.
So it sounds like antibodies are very specific in terms of what they attach to. In comparison, how specific are the actibodies? Because the key lock analogy, there are certain things that are like skeleton keys that can open multiple locks. So I'm just wondering how comparable is the actibodies versus the antibodies in terms of how specific they are?
So we can actually design them to be more specific to certain things, meaning they bind stronger, or we can design them to bind weaker. So there are actually antibodies that bind fairly well to one thing, but will bind to something else that kind of looks like it too. So we can make sure that depending on the application, we design it so it binds very specifically. And yeah, we have that in our power.
I'm just curious with the antibodies, proteins basically, and the stability of proteins, unfolding, folding, whatever, which just getting back to when I worked in plastic bottles. They like to stick to polar things, so you end up with you can't find the drug because it's stuck to the wall of the bottle. But the stability of the protein is dependent on the environment that it's in, the pH, whatever. Is antibody one more stable, more potent to stability than the other, long term, for obviously shelf life of drugs?
Very good question. So antibodies are made from mammals, so in some ways their shelf life might be a little lower depending on what your intended use is, but because they're made from mammals, they're far more useful for us as humans. So in terms of being more stable in our environment, I think antibodies currently will be more stable. Now our hope is that when we design these actibodies, we can make sure they're just as stable in our body, especially since they're so much less expensive and faster to produce. So it all goes into us being able to engineer it so that it's friendly in the human body and just as stable.
You listed a couple of other benefits to the actibodies besides just improving diagnostic tests, and I was wondering if you could elaborate on one of those. I don't remember what they were, but like helping medicine in our work in our bodies, like that maybe?
So the question was, what are some other benefits of using this other than like diagnostic tests? So one very popular use of antibodies and therefore actibodies would be to help make better drugs, better targeted drugs, meaning they only attack, say, just the tumor cells and not your normal cells, for example. So actibodies could be hugely helpful in that, and they could also help better release the drugs when you need it to. So again, improving targeting. Another use actually is more in research. So the kind of research that Stella, I, and Vinayak will do, a lot of biology research involves using antibodies, these sort of binding things. And if you have an actibody that has a natural capture and release, that can make a lot of the work we do a lot easier. So we would appreciate that. Okay, thank you very much.